Hello again, this is Layla, and in today's video we're going to be talking about the recombinant DNA. We're going to be discussing 3 points. The first one is what is a recombinant DNA. The second is the steps of making a recombinant DNA, and the final one is the uses and aims of recombinant DNA. Okay, so let's begin with the first point. What is a recombinant DNA? So, recombinant DNA or are DNA molecules. They're basically DNA molecules that are made in laboratories through a process called genetic recombination. Okay, I know this is kind of confusing, but we're just going to say the meaning of genetic recombination, then we'll define recombinant DNA in a more comprehensible way. Genetic recombination is a process through which the genetic material is exchanged between different organisms, which results in an offspring containing different genetic traits from either parents. So, in order for us to make a recombinant DNA, we're going to have to take a DNA fragment from an organism and then another DNA fragment from a completely different organism, and then when we join them together, this will form a recombinant DNA. Okay, since we have defined a recombinant DNA, we can conclude that the difference between the normal DNA and the recombinant DNA is that the DNA is found naturally in organisms, while the recombinant DNA is made in laboratories. Since the recombinant DNA is made from different sources, let's clarify the idea through the second point that we're going to be discussing today, which is the steps of making the recombinant DNA. Okay, so the first thing that we have is the plasmid. A plasmid is a small, circular, double-stranded DNA molecule that is usually found in bacteria and yeast cells. Then the restriction enzyme bonds with the plasmid. A restriction enzyme is a kind of protein that bonds with DNA molecules so that it cuts the DNA fragment. Okay, then we get another DNA and we just specify the part or the gene that we want. Which is the gene of interest. Let's say that this gene contains insulin and we want this insulin so that we can manufacture later for the treatment of diabetics. So again, the restriction enzyme bonds with the gene that we want and it separates it from the main DNA. What happens next is that both DNA fragments are supposed to be joined together. DNA ligase is used in joining the two DNA fragments chemically. In the end, a recombinant plasmid DNA molecule is formed. Okay, now that we have discussed the definition of recombinant DNA, and then we talked about the steps of making recombinant DNA, we're going to talk about right now the usage of recombinant DNA technology. So, recombinant DNA can be used in the studying of individual genes and in the treatment of certain diseases. I'm going to clarify a usage for recombinant DNA technology through a process called gene cloning. Gene cloning is the process through which we can make large quantities of recombinant DNA. So I'm going to use the recombinant plasmid DNA that we have discussed previously. So here we have the recombinant plasmid DNA, but we're only going to add an antibiotic resistant. Then we're going to have a solution. This solution contains bacteria and we're supposed to add the recombinant plasmid DNA into the solution. So basically what happens is that when we add the recombinant plasmid DNA into the solution, some bacteria, they take in the plasmid and some bacteria don't. The process through which bacteria cells are mixed with recombinant plasmid DNA is called transformation, and this takes place or it happens using electric pulsation or a heat shock. What happens next is that cells start replicating and the bacteria cells that contain the recombinant plasmid DNA, they begin making copies of it during cell replication. What we're going to do next is that we're going to add the bacteria in this plate, and this plate is going to contain nutrients for the bacteria to grow on and antibiotic. Okay, this is why we added an antibiotic resistant in the recombinant plasmid DNA in the first place. because only the bacteria cells that took in the recombinant plasmid DNA, which contains the antibiotic resistant, are the ones that are going to survive on the plate and the ones that are going to start colonying and growing in the plate. That's all for today's video. Thank you.